Hello, everyone. Thank you for attending this LabRoots webinar sponsored by Beckman Coulter Life Sciences. My name is James McCracken, and I am a senior product manager with the Biodiscovery Flow Cytometry business unit. And I'm happy to introduce our speaker for today, Tamar Tak. Dr. Tak is currently the coordinator of the Flow Cytometry facility at the Leiden University Medical Center in Leiden, the Netherlands. And he's going to speak to us a bit today about his shared resource lab's perspective on the CytoFLEX SRT cell sorter and what his experience has been like integrating that within a multi-user environment. Very much looking forward to this. Thank you very much, Tamar, for agreeing to speak with us, and please take it away.
All right. Thank you, James. So first of all, the disclaimer to say that this is all my opinion and not that of Beckman Coulter. So as James mentioned, I'm located in the Leiden University Medical Center, which is in Leiden in the Netherlands. So it's a university medical center for research, education, and patient care, and it really has a very large academic portion. So a lot of research is being conducted there. And what I think is really nice about the hospital is that it's really closely situated together with the university and also with the Leiden. It's in the Leiden Bioscience Park, where there's a lot of the larger and also large biotech companies, but also quite a few smaller startups. So the facility is a very big one. It's established in 2017 and currently houses 24 instruments. So it's a really wide range of instruments, cell sorters, analyzers, both spectral and conventional. And we also have a number of mass cytometers in the lab. And these instruments are used both for research and for diagnostics. So on average, every year we see about 500 unique users. And in total, they book, at least last year, they booked more than 20,000 hours of sessions. So most important part of the facility is not the instruments, but it's a team. Marjolein is the operational manager. Simone, Deborah, Jacqueline, Ruben, and Inye are the current operators. And IJsbrand, he recently left the core, but I still want to acknowledge him because he did a lot of work on the CytoFLEX SRTs. So what do the operators do? Of course, they make sure that the instruments are performing as good as possible. So every day they do the instrument startup and QC, and if necessary, they do some troubleshooting to make sure the instruments are working like they should. Of course, we write standard operating procedures, and during the day when the users come to use the instruments, we always make sure that there's an operator available in the lab for user questions and some troubleshooting if necessary. Of course, we train our users to use the instruments themselves. So last year, that meant that we gave about 300 training sessions. We organize user group meetings to bring users using the same technique together. So they're often from different departments, but we like them to learn from one another. So that's why we organize these user group meetings. On top of that, we organize seminars on special subjects, and we perform operator-assisted cell sorting. And that last bit is what I really want to talk a bit more about today. So for the operator-assisted cell sorting, usually a customer designs their own experiment, and this can be with or without help of the FCF. When they want to perform the experiment, they can book a slot in our commercial booking system, and the system asks them to fill in a sort request form. And in that form, the user will have to give information about the biosafety, which fluorochromes they want to use in their experiment, the collection format, the time they think they need, the number of samples, number of cells, etc. Then the operators will check that sort form and see whether the user really will have enough time and whether they selected the right instrument for their experiment. Once it's approved, the user comes to the facility, and then the operator will set up the sort. So they'll do the QC, set up the gains, set up the stream, drop the delay, and the whole sort setup. And then together with the user, they'll do the gating. Well, once everything's set, the operator will perform the whole sort. So they'll monitor the sort, do the purity checks, and do the data export. After which the user gets their cells, the users get their cells, and they can do follow-up experiments. So this is, of course, very nice for the users, but it does take quite a lot of time of the operators. And because we don't have enough operators to work in shifts, we can only offer assisted sort during office hours. So we can't offer any sorts outside of office hours, and because we don't have enough assisted sort time available, sometimes users have to wait for weeks before they can actually do their experiment. So that meant that we wanted users to be able to perform their own sorts. But the instrumentation that we had at the lab, in the lab, wasn't really easy to use. Was more than 10 years old. So this was not really equipment that we wanted to train all of our users on. So that meant that we had really a need for easy to operate cell sorters that can be used by users without operator assistance. Luckily, our institute gave us budget to purchase these instruments. And for the purchase, we had to go through a public tender procedure because that's the European rules. And so to start that tender procedure, what you do is you have to publish a list of requirements. And then companies can actually say, well, we can fulfill these requirements, yes or no. But first, we had to determine what our requirements were. So we did that by sending out a user questionnaire where they could fill in what they were currently doing in experiment-wise and what type of experiments they would like to do in the future. We, of course, consulted our advisory board. We gathered together operator experiences, and we performed an analysis of the usage in the preceding years. Because those short forms that we ask our users to fill in, they actually give us a lot of data on the experiments that they're doing. So what we got out of the results that we got out of that was that our users usually use 85 micron nozzles. They have very small panel sizes, so most of the sorts are only with 0 to 3 fluorochromes. Users usually sort only 1 or 2 populations. They do use both tubes and plates that they want to sort in, and they have a wide range of sample and collection temperature. So that means that most of the sorts that we were performing were actually fairly simple. So it's only 1 or 2 populations and a few markers. So and on top of that, we analyzed the sort request form to figure out what the configuration of the instrument would have to be. Because some lasers and detectors were not used all that often, whilst others are used quite a lot. So it's mostly the things like BV421, BV510, FITC, PE, PE size 7, and all of the red fluorochromes, excited by the red laser. So this meant that we didn't really need all that many fluorochromes, but we did see that there was an increase in the use of fluorescent proteins that require a yellow-green laser, such as M. scarlet and TD tomato, which are excited much more efficiently by that laser. So a big wish for us to get an instrument with the yellow-green laser. So taken together, that led us to make a list of our minimal requirements. So we needed an instrument that could sort at least 2 cell populations, detect at least 8 fluorescent parameter. We wanted it to sort at a speed of at least 10,000 events per second and be able to sort in tubes, 96 well plates, and be adaptable have adaptable sample and collection temperature. So that was a hardware requirement, but because we really wanted our users to be able to operate the instrument itself, we also put a lot of requirements on the software. So we wanted an intuitive user interface, an automated workflow, and do some easy troubleshooting and be able to do to troubleshoot the instrument easy, and do the maintenance should be easy as well. So that was a minimum requirements. But all the eligible instruments were also, so we didn't want necessarily an instrument that fit the minimum requirements, but if we could get something better, well, we would very much like that. So all the instruments that were eligible, they were graded on their technical specifications. So they had to meet the minimum requirements, and then they could get bonus points if they performed better than the minimum requirements. So for example, instead of doing only 2-way sort, a 4-way sort would give extra points, a low carryover, the option for additional sort formats, such as 384 well plates, a higher speed or efficiency than the minimum requirement, and a larger amount of fluorescent channels. So that would amount to about 30% of the total points that the instruments could get. Now, after that, so that was based on technical specifications only, but we also performed an on-site test. And in that on-site test, we really determined the carryover, sort speed, and the purity, which was only 10%, but a really large chunk of points that the instruments could get was based on the ease of use. So it was ease of use of the hardware, ease of use from the software, like how intuitive is it, how easy can users learn it in just a few hours. And we tested this actually by having a lot of our operators test the instrument after a short training, but we also asked both experienced and unexperienced users from our core facility to try the instrument and see whether they were able to perform a sort. And then, of course, the remaining 30% was on price. So after all this testing, so we tested multiple instruments, and then actually the CytoFLEX SRT received the highest user testing score. And that's mostly because the users thought the software was fairly intuitive, the workflow was easy, it was not that hard, it was difficult to make really mistakes in your experiment. Data import and export was easy, and the startup and shutdown were both very easy to perform because there's step-by-step wizards that guide you through it, and also the startup and shutdown time was pretty quick. Besides that, so switching of sort formats was easy, there's different sort modes, and as said, the troubleshooting was fairly easy. And what we really liked there was that the manual is very complete, and if you ever get an error message, it will actually give you a code, and you can just find it in your user manual, and then you'll get solutions to what you can do to fix the error. So the conclusion was that it was very easy to use, and it requires very little time to train someone as long as that person has some experience in flow cytometry. So some examples of that. So there's some tools that you get. So there's a lot in the SRT actually has a lot of cameras in the system, which will monitor the stream. So here you can see the stream that is monitored all the time, but also the sort is monitored. So if there are cells going through one tube, you'll see this in a screen. But if there's cells that actually don't make it, or there's the stream is spraying a little bit, you'll get an error message. And I think that is something that we didn't have in our older instruments. On top of that, there's some nice features such as, well, you can really follow the sort, but for example, the sorting will stop if your tube is full. So those things we really liked. And then in the end, this resulted in us acquiring 2 instruments at the FCF. So they're both 4-laser instruments capable of detecting 15 fluorescent parameters. One of them was placed in our BSL-1 lab, so that is outside of a biosafety cabinet, but we've also got one in a biosafety cabinet, and that is placed in a BSL-2 lab. So that meant that we could actually finally move from an assisted to an autonomous sort. And we also, and the workflow for that would be a lot more easy on the operators. Because again, so this is the operator assisted sort where the operators perform most of the instrument setup and the sort itself. And then we move towards the to the autonomous sorting. There, the user would do most of the settings themselves. So it would still start up the instrument, and we would still check the sort form and give that an okay or not. But besides that, the idea was that the user would do all of the tasks themselves. So some of the first things that we did after we acquired the instrument, so we had to start training the users and making sure that they would be able to use the instrument themselves. So a very important first step is to condense the 730-page user manual into a very short 6-page daily use standard operating procedure. So it's a lot shorter, but it does deal with things like instrument startup, instrument shutdown, how to set up your experiment or compensation, and also some very basic troubleshooting is in the SOP. So besides just giving our users an SOP, we actually give a lot of trainings for our users. So we do ask our users to have at least some experience in flow cytometry. So we ask them to have about 3 months of flow cytometry experience. So we'll want them to do normal training first, use the instruments a little bit, get used to how flow cytometry works, do some gating, do some experiments, and then they can start doing the training. And then the training consists of a theory session. So it's in a classroom setting of about 2 hours, where we really explain all the principles of cell sorting. We give an overview of the hardware, we show how the software works, and we also discuss the best sorting practices and some sample preparation. And then the next day, in groups of 2, our users will get a hands-on session where they can actually do a small experiment on the instrument. So again, we give a bit of an explanation on the hardware. We explain the biosafety rules, but we actually have them do the instrument startup and shutdown and perform a sort with beads. So it's a very simple experiment that we have them do. So it's only a 4-color panel with compensation beads, but at least it gives them the opportunity to perform a small experiment. So we have them do start up the instrument, do the QC, acquire a sample, perform some compensation and gate their cells, and actually do a sort of these beads. So they'll have to do a sort calibration, and we ask them to sort in tubes and in plates both, so that they've tested both of that. And they'll have to do the system shutdown and really do the daily maintenance of the instrument so that we can be sure that they can give us a nice and clean instrument. So in the years that we've had the instrument, we've trained 146 users so far, and we group these actually in 3 competence levels. So once you've done your training, you'll get the competence level A, which means that you can sort between office hours, but we make sure that there's always an operator available to troubleshoot the instrument if you run into any trouble. Level B you can get after you have 10 hours of experience, and they'll do a short competence check just to see that you can really handle the instrument well, and we'll go through the system startup and QC again and do some more troubleshooting training, so that if users want to sort outside of office hours, if they run into trouble, they can really operate the instrument independently. So level C, it's a bit the more rare one. There people can also sort the weekends and get during the training and the competence check, they get some more advanced troubleshooting training. So far we've trained 146 people. 90 of them are right now in competence level A. We have 14 people that want to use the instrument outside of office hours, and so far we've only had 1 person that felt the need to sort in the weekends. So so when the users started using the instrument, so this was what I showed you before, the expectation of the autonomous sorting workflow, but in fact we found out that a lot of our users actually did need a lot of help during things like the sort setup and the sort itself, as well as the instrument setup. And it doesn't necessarily have much to do with the instrument, because we still think that the sorter is easy to use, but it does turn out, which as an operator, we actually knew that cell sorting has different requirements than just normal flow cytometric analysis. And on top of that, we had some users that did lack some experience with conventional flow cytometry, or at least they were making some mistakes that they would also be a problem with just analysis. So for example, we saw people setting their gates wrong. So they were setting very broad gates, where with sorting, you really want your gates to be very nice and tight around your population. We've seen people making errors with axis scaling. And here is an example of something we encountered with our user, that they were trying to sort everything except this population on the top left. But in fact, they were missing a lot of cells, because a lot of cells were actually on the y-axis, on the x-axis. And then when we showed them how to do the scaling properly, it turned out that they were missing a lot of cells there. Something else that goes wrong is the sort precision, so that they don't know whether to use purity or yield. And what most of the problems that we see are due to sample preparation. So people are trying to sort something with a lot of clumps, and they're very clumpy cells, so that the instrument gets clogged a lot, and then they run into trouble as well. So after we noticed that, we started to really highlight all of these things during the training. So now people will talk about how to set the gating. We'll actually have them do their axis scaling. We'll explain the sort precision again, and we really included a lot longer session during the lecture about sample preparation. On top of that, we made a document, sort recommendations document of a few pages where we give some advice on which buffers to use, which precisions to use, how to treat your sample, how to process your sample, etc. And we put that on our facility website, but we also put it next to our instrument because users tend to be bored when they're sitting there. So we hope that they start reading it. And on top of that, we are starting to offer assisted experiment setup for the first sort. So once people have done their training, the first time they want to use the instrument, they can request an operator to help them, and that's free of charge, just so that we can make sure that their first experiment, the setup is all done correctly, and we can give some more advice. And that actually helped improve the quality of the sorts a lot. And then lastly, we actually revoke the booking rights if someone hasn't used the instruments for 6 months, because we see a lot of people that they take the training, but then they never actually use the instrument. And then maybe a year later, they want to use the instrument. They've completely forgotten how to do that. And then they run into trouble and blame the machine on not working correctly. So after 6 months, we revoke their rights. If they want to get back those rights, they either have to follow the training, or we do a short competence check and if they can show that they can still use the instrument, then they're good to go and use it again. So what is the reality now of the instrument usage? So we had the instrument since 2021, and both of the instruments are actually used quite a lot. So they're used 3 to 5 days per week, and we get approximately 100 hours of usage a month. And we can really nicely track that through our booking system, because there's, we can just see for every month, for every booking, we can see which instrument was used. And we also been able to really have a look at like what type of users do we have? Is it, do we have a few super users, or is it mostly incidental use? And in fact, it's actually mostly incidental use. So most of our users have only used the instrument for, well, maybe 1 to 25 hours. But we also have some super users who have used the instrument for more than 100 or 200 hours. We try to have a look, whether these instruments are used the same as our old instruments. So do we see more or less fluorescent parameters? And you see that for a lot of the sorts, people only use one, 2, or 3 fluorescent parameters. So a lot of that means that people will be sorting only on GFP or another fluorescent protein. But we do have some users that actually push the instruments a bit more to their limits and sort more than 10 fluorescent parameters in one experiment. So from the sort form, we can also see what people use their, so what type of samples people are sorting and what they use the sorted cells for. So it's a research hospital, so a lot of the samples that you'll see are PBMCs. But we also see a lot of cell lines being sorted, and there's a huge chunk of stem cells, either induced or embryonic, and approximately 10% of the samples that go through the instrument are tissue samples. So these are digested tissue samples, either from mouse or human. And what people use the cells for that they sort, well, most of it is cell culture, but there's also quite a few functional experiments, PCR, DNA or RNA sequencing, microscopy, and there's even one user who uses the samples for NMR analysis. So we have 2 instruments, so one at BSL-1 level and the other is at BSL-2 level. But when we look at the sort forms, we actually see that most of the samples that are being sorted, they do not contain pathogens or GMO. And that is both on the instrument sort that is placed on BSL-1 and BSL-2 level. But we do get a lot of lentiviral transduced samples. And for the BSL-2 level, we get retrovirus transduced samples. And that is one of the main reasons to include a BSL-2 instrument, because the retroviral transductions are done a lot. And for that, in our institute at least, we need the instrument to be at BSL-2 level. And recently, in the last year, we're starting to see requests for people that want to sort bacteria. And these we also do at BSL-2 level, and that's mainly because of the safety cabinet that the instrument is placed in. Then the collection format. So the SRT does actually offer a range of collection formats. So you can use 15 ml tubes, 5 ml tubes, 96 or 384 well plates, and these can be either normal or deep. And you can even sort on tissue slides. But in fact, if you look at what people are using, it's really mostly 15 or 5 ml tubes, standard well plates, and there's a few people that sort of, very rarely we get 384 well plates or other plate formats. And we've never seen anyone sort in a deep well plate, and we've never seen anyone sort in a microscopy slide. But no, maybe in the future people will start using that. So after having used the instrument for more than 2.5 years, we do see some issues that occur quite frequently. And that I also want to discuss a little bit. So what we are seeing is that the instruments appear to be fairly sensitive to temperature changes. So if the temperature goes up or down too much, you get a warning that you need to rerun the QC. That's not necessarily a problem of the instrument, but it's a problem of our lab that can be, especially the BSL2 lab, is fairly small and can heat up pretty quickly if all the instruments are running. So it's actually quite nice that the instrument will tell you that you have to rerun the QC. We did see from time to time that the deflection plates got wet because the sample was spraying a little bit, but that was mostly solved by a new nozzle. In addition, and I'll show you a solution for that as well, we ran into quite a bit of pressurization problems during startup or sort calibration, and there's a lack of crash detection on the probe that also needed a little bit of improvement from our side. But first off, the pressurization issues. So that is something that the sheet tank is not, the pressure on the sheet tank is not as high as it is in some instruments. So sometimes during the startup of the instrument, we get a pressurization error, or sometimes it would occur a little bit later in the sort setup, so when you were setting up the streams. And it was not always clear from the error message that the pressure was a problem. But we found that if we tightened the lid of the sheet tank or lubricated the O-ring in the sheet tank, that helped a lot and that prevented a lot of problems. So we started to tighten the tank more and more until some of our users were not actually able to open it anymore. So that was not an ideal solution. But then one of my operators actually came with the idea to just add a second O-ring. And I thought that was never going to work. But in fact, adding a second O-ring on top of the original one, since we've been using that, where we have very few pressurization problems. So if you also see these trouble, I would really recommend to just add a second O-ring, and that helps a lot. So regarding the crash detection or protection, so I'm not sure if it's a bug or a feature, but the tube holder where you place your sample in can be tilted about 15 degrees. And that actually does make it a little bit easier for you to load your sample. So in the first few trainings that we gave, we taught our users that you can do that and that it's easier to put your sample in like that. However, there is a risk of the sample probe getting stuck between the tube and the holder. So here is an image of that. So here we got the tube is being tilted and the probe just slipped past the tube and between the tube and got stuck between the tube and the tube holder. So the probe should, when it's going down, it should slip through the fitting at the top when it's encountering resistance, but for some reason it didn't at this time. So maybe it's because it didn't encounter enough resistance, or alternatively, one of the operators just tightened it too much on the top. But this was fairly problematic because we really had to use a saw to take the tube out, and the probe was damaged beyond repair. So then we really started to teach our users to really not ever tilt the tube and always check if it's tilted or not. But we still had some people, we still had this happen occasionally. So what we did, luckily we have a very good workshop in the LUMC where they have 3D printers, and they designed for us a very small insert that just fits between the sample holder, the tube holder, and this metal ring that you see here, and that now prevents the tubes from tilting. So the users just can't tilt the tube anymore. So that really, since then, we never had any probes being damaged anymore. So if you're interested in this, just send me an email and you can get the cut files to print it, or yeah, it's not that difficult to make. So you can even ask your own workshop to print one for you. So on top of that, so we do have a very small wish list on the instrument. So what we would like to see is a 130 micron nozzle. We only have a 100 micron nozzle right now, and for very large cells or very fragile cells, a 130 micron nozzle would be very beneficial. The larger sheet tank does exist. So with the one that we have, we can sort for 11 hours, which is almost always enough, but there's always that one user that wants to sort for more than 11 hours in a row, and then you have to refill the tank during the experiment, which is not ideal. So as I say, so the tanks do exist now, but we haven't gotten them in our facility. So some of our users, so we have an instrument with 5 detectors in the violet channel, so they can detect 5 fluorochromes, and some of our users always want to use one fluorochrome or another that isn't present, that we don't have the filters for. So if it would be possible to have more detectors on violet, that would be great. And lastly, something that we would like is to be able to sort out of 15 ml tubes or Eppendorf tubes. But I can imagine that the 15 ml tube is never actually going to fit. So taken together, I believe that the CytoFLEX SRT at our facility is very well utilized. Most of our users praise it because of its easy to use software. And for us, it was really beneficial because it allowed us to offer autonomous sorting and allow sorting after office hours, which is very good for our users that have patient material that comes at strange moments. So to move towards the autonomous sorting, it only requires a few hours of training. So it's really just 4.5 hours of training that people get on the instrument. But you do need to train your users on good sorting practices. And some of the users did turn out to need a refresher on basic flow cytometry. But that is why we're now helping them during their experiments and helping them to set up their first sorts. And then that brings me to my last slide, so the most important one, because of course I got a lot of help from all the operators, Professors Frank Staal and Jacques van Dongen, who set up the beautiful facility that I'm running right now, and the people from Beckman Coulter in the Netherlands, who have been of great assistance to help us with our instrument. And I would like to thank you for your attention, and I would be happy to answer any questions.
Okay, great, Tamar. Thank you very much for that. I have seen a few questions, and I do have maybe one of my own. I'll start with. You definitely have a robust training program there, and I think that's very admirable and important for adoption of new technology like this. Do you find your users are pretty enthusiastic about learning to support their own sorts? Do they lose any enthusiasm when they see the time that might be involved to support their own work, or do they remain pretty enthusiastic about it?
We see that most of the users are actually pretty enthusiastic. I think so. There will always be some users that prefer to have operator-assisted sorts, and I think there's still really a place for the operator-assisted sorts. So if it's a very complex sort, or if it's a user that only wants to use the instrument once a year, you don't actually want to train that user. So yeah, so I think there's still a reason to do operator-assisted sorts. But a lot of our users, and especially the super users, they're really enthusiastic about being able to do that themselves. People running precious patient material that comes in at odd hours, they're very happy to use it. And I think so, and actually the time it takes for the training, so it's 4.5 hours, it's exactly the same time that they would have to spend if they do a conventional or a spectral course.
All right. And maybe I just didn't notice this as you were speaking. Do you also teach users when talking about sample prep and things like this, how to estimate how much time they need to book in order to complete their sorts?
Yes, we do. So we teach it during the training, but also at the top of the sort form that they have to fill in. We give them an estimation of how many cells they can run
Mm,
per hour, and then they can estimate themselves how much time they'll need. But for every sort form that they fill in, we actually check how many samples they have, how many millions of cells they want to sort. And if we think, well, they're never going to make that, we'll send them an email just to tell them, like, well, I would recommend you book a few more hours.
okay. Okay. Very good. Um, Maybe you could tell us maybe some of what the most common issues that your users need live help with, right? You always have someone in the lab, but what are the common things that people still need assistance with?
What they really need assistance with is usually when, for example, the sort monitor says when the auto maintain switches off.
Mm,
So the stream is being auto maintained, and auto maintain suddenly shuts off. Then the user will come to us and ask us, like, well, what could the problem be? And most commonly is that they've done a sort calibration, then they set up their whole experiment, and maybe it's already 2 hours later. So, and then when they actually start performing their sort. So they really do the sort setup way too early. So that is a very common issue. And the other issue is sample prep. So there's too many dead cells in there. There's too many clumps. Yes, and especially with a lot of dead cells, the efficiency goes down. And with clumps, well, you get clogging of the instrument. And that we still see a lot, despite teaching them, well, despite giving them a lot of advice on how to treat their samples. Of course,
okay.
sometimes a user is just unlucky, and the sample is of poor quality.
It happens, right? Um, I wonder, do you and your staff have any pre-tested staining panels that you offer to users for certain applications, or do you just assist them into making things particularly for their needs?
No, so we don't actually offer any standard panels, but we do know our users quite well, and we do know what they're working on. So we usually recommend them, oh, you want to do this staining on this cell type, go talk to that person, or
Mm,
go talk to the other person. And I think that is a benefit of those sort forms, because we can just look up someone else who's handling the same type of material.
and your users can kind of self-support each other in that community.
Yes, well,
That's
when
very cool.
we do try to keep involved in that, but there's so much knowledge and there's so many different cell types that people are working on, T cells, B cells, myeloid cells. Well, I'm not an expert on all of them, and neither are my operators. So quite often it's better to just tell them which other user has a lot of experience on it.
Mhm. And then maybe there's one last one I'll put in here, which is I see, of course, that you have a wide variety of different kinds of hardware in the lab. How would you advise someone that maybe has a panel they built on a spectral instrument that finds a population that they then want to sort? How would you advise them to approach that?
It really depends on the panel that they have, of course. But usually, a panel that you use for analysis is not something that translates completely to a cell sorter. For analysis, you're not as strict. The separation doesn't have to be as good as for a cell sorter. And what we see a lot with spectral people designing panels for spectral instruments is that the lineage markers, they're usually on very rare fluorochromes. So those are not necessarily the ones that we can detect with our cell sorters or with any of our conventional instruments, actually. So sometimes it's possible, but quite often users will have to redesign a panel. Or if it's possible, they can, of course, design a panel that will work both on a cell sorter and on a spectral instrument, but then they'll have to really design it like that from the start.
Hmm. Hmm. All
Of
right. Sorry,
And of
something else?
course, of course, we would be very interested in getting a spectral sorter.
Understood. I'll add that one to your wish list. Thank you, Tamar.
Yes, let's do that.
All right. Well, I think that's all I've seen come in. I'd like to thank everyone again for your attendance and your attention. Thank you very much, Tamar, for agreeing to present what your experiences have been like. And thank you to LabRoots for broadcasting this. And I'll see you next time. Thank you very much.
All right, thank you.